Thank you for inviting me back again tonight. And thank you to Bill and the entire Park family for continuing to organize these events and push forward the Miles for Mary efforts to fight brain cancer. I heard the second annual walk was really a great success, so it's wonderful to hear. I think it's an amazing statement about the impact Mary had on everyone around her and the continued efforts of the Park family to raise awareness about brain cancer. I certainly enjoy coming to these events because I'm very passionate about the research that we do, and I hope to share that excitement with all of you because you are a very critical part of this work. And your contributions have really sustained our research efforts and made it a reality. So hopefully you too can really feel proud about the advances we have been making in fighting this challenging disease. So what I thought I would talk specifically about tonight is the project that the Miles for Mary effort has been supporting. But first, let me lay the landscape again for really what the challenges that we're facing. So glioblastoma is fairly rare in the landscape of cancers, and it's a primary brain tumor, meaning it comes from the brain itself. It's not a type of cancer that starts someplace else in the body and spreads to the brain. And there are only about 10,000 cases each year in the United States. And so that's very small compared to something like breast cancer or lung cancer, and that creates a lot of challenges for us as researchers in obtaining funding to do research and obtaining increased awareness about this type of tumor. And so the way we treat patients is with maximum safe surgical resection. So we try to get out as much of the tumor as we can safely. And then we follow that with a combination course of radiation and temozolomide chemotherapy. And that temozolomide chemotherapy can go on for up to 12 months after initial surgery. Unfortunately, though, despite this very aggressive treatment, the median overall survival is only about 15 months. So that means that 50% of patients don't live 15 months, but 50% of patients live longer than 15 months. Although when you start talking about 2-year survival, it really gets closer to 10%. So it's really quite a devastating disease for a patient to have. Temozolomide, the chemotherapy that we use for this type of cancer, was FDA approved in 2005. And then bevacizumab, which is a drug that's supposed to target tumor blood vessels, was FDA approved. This was our newest drug that we have in our armamentarium, was FDA approved in 2009. However, there were 2 recent large randomized phase 3 trials adding bevacizumab to our standard surgery, radiation, Temodar chemotherapy course, and that unfortunately only improved median overall survival to 16 months. And so clearly very little has changed in the last 10 years since Temodar was FDA approved, and we have a lot more we need to do in order to talk about or fight this kind of cancer. So one of the projects that I have been working on is looking at the role immunotherapy and more broadly inflammation plays in these tumors. So these tumors are very smart. They suppress the immune system, so the immune system does not recognize the tumor as foreign, and it does not fight off the growing tumor. And so the goal of immunotherapy is to reawaken the immune system so it starts to attack the tumor in the same way that would attack a virus or a bacteria. As most things in cancer, this problem is much more complicated than it sounds. That idea is very appealing to get the immune system to wake up again. The challenge with immunotherapy is that the response to treatment can be very difficult to assess. And the standard way that we measure brain tumor response to treatment is by using MRI scans. And you look at how much contrast dye the tumor is taking up when a patient undergoes an MRI scan. So what you're seeing here in this picture are 3 MRI scans of a single patient during treatment with an immunotherapy. The patient is looking straight out at you, so looking at each one of your eyes, and these are vertical sections through the patient's brain. And so what you see, this is before the patient starts treatment, and there's a little bit of uptake of the contrast dye around the surgical cavity. 5 weeks into treatment, there's this really robust uptake of contrast dye. This patient ended up staying on immunotherapy, and you see actually that contrast dye has gone away over time. But the challenge is how do we interpret this time point in therapy? Because when you're looking at this MRI scan, when I'm looking at this MRI scan and I have the patient sitting across from me in the room, I don't know if this uptake of contrast dye is actually active growing tumor or is it inflammation from the immunotherapy. And I want that inflammation to happen because that means my treatment is working and it's fighting off the cancer. But this is the challenge. I don't know, and it's very hard to tell a patient, well, let's just sit and wait and see what happens in a month from now. That's sometimes a hard sell for patients to just sit and wait. This challenge is not just restricted to immunotherapy. I should also say it has broader implications. And so what we've learned is that there's a subset of patients who are just treated with standard radiation and temozolomide chemotherapy, who also, in the few months after getting that combination chemotherapy and radiation, will also have this very robust inflammatory reaction to that treatment. And you still don't know in that subset of patients, do you sit and wait or do you change therapy? Because you don't want to let the tumor continue to grow, but you don't want to change a therapy that's potentially working and having the effects that you want it to do. Next slide, please. So what we're trying to do with this project is to figure out, is there a better tool than just routine MRI scans to measure inflammation in tumors? So what we've done with the Miles Fumeri support is to design a clinical trial. And our clinical trial has two arms to it, so two cohorts of patients. The first cohort in arm 1 are patients who are treated with immunotherapy. They have their routine brain MRI to assess their response. We have to do that because that's our standard way that we assess response. If the treating physician determines that that's a response, they're going to continue on immunotherapy. If the treating physician determines that that's tumor progression, then they're going to change treatment and do something differently. What we're going to do and add into this trial is to do a special technique which is called a PBR PET. And PBR is a new PET tracer, and so PET is another way of looking at tumors. It's not an MRI scan, but it looks at tumor metabolism and looks at different molecules in the tumor. And so PBR is uptaken into areas of inflammation. And so what we're going to do is do a PBR PET scan prior to the start of the treatment. That'll be our baseline scan. And then we'll do another PBR PET at the same time as the MRI scan is done. And in that way, we hope that it'll help us interpret what's going on with the MRI scan. Meaning if we see a lot of uptake on our PBR PET scan, then that's going to be more consistent with inflammation. If we don't see a lot of uptake on our PBR PET scan, that's going to be suggestive of active tumor because the active tumor is working to suppress the inflammation. That's how it evades the immune system. Now, these are not real PBR PET scans because we haven't started this trial yet, and I'll tell you in a minute where we are, but this is the idea and the concept of what we're doing. And then the second arm of our trial is going to be looking at that other cohort of patients that we know have this very robust inflammatory response to standard chemotherapy and radiation. And so it's a similar idea. They'll get the standard chemotherapy and radiation, get their MRI assessment, and there has to be some clinical judgment as to whether or not the patient is responding or not responding. But at the same time, we'll get this PBR PET scan and see if that can help us interpret the MRI response and help us figure out if patients are actually having true tumor progression or if this is the inflammatory response that we like to see to therapy, meaning that the immune system is helping us to fight off the tumor. Next. So there's a lot of steps that go into generating the design of a clinical trial, and this is why it's taken us a long time to reach the stage that we're at. So when you're designing a clinical trial, the first step is to apply for funding. You have to pay for your clinical trial somehow. Next, you have to generate what's called a protocol document. And this is a 60 or 70-page document that outlines exactly how the trial is supposed to be run, who's going to be eligible for your protocol, how are you going to select patients, what's going to happen if a certain toxicity or problem comes up, how are you going to analyze your data. All of that has to be clearly spelled out ahead of time. You also have to write the informed consent document. This is what the patients are given in order to sign when they want to participate in a clinical trial. So once you have your funding and you've written your protocol document, you have to submit it to the disease committee. This is a local MGH committee that is staffed by the neuro-oncologists, and the neuro-oncologists say, okay, your protocol sounds interesting. We will open that trial at MGH or Dana-Farber. We're a virtual cancer center. At this time, you might also have to file an IND, an investigational new drug application with the FDA. So anytime you're giving a patient a new drug, the FDA has to be involved. Once that's done, you then submit it to your scientific review committee. This is, again, a local institutional committee that looks at your protocol document and really looks at the scientific rigor of it. Are you going to be able to do this study and answer the question that you're actually trying to answer by conducting this study? Once the scientific review committee has reviewed it, then you have to probably do some revisions, and then you can submit it to the Institutional Review Board, or IRB. The IRB is really focused on patient safety, so they really just want to make sure you're not going to harm patients with your clinical trial. And then finally, once all those steps are done, then your protocol can open. You have to register your protocol on clinicaltrials.gov. This is a central repository for all clinical trials in the United States. Also, international studies are often registered here as well. We now also have to report trials, particularly if they're NIH funded. You have to report the results of the trial on clinicaltrials.gov. And then as the protocol opens, then there's serial paperwork and regulatory reports that you have to submit back to the IRB and back to your whoever is sponsoring it, the FDA, these people that really want to make sure that your trial is being run in the proper manner. So where are we at this point? We've applied for funding, so we got an internal MGH grant to do this trial, and then we're also leveraging the Miles for Mary support in order to do this, because our internal MGH grant does not support all of the costs for the trial. We've generated our protocol document and consent. We've submitted it to the disease committee. We filed an IND because our PET tracer, since it is a novel tracer and it's not FDA approved, we have to work through this internal mechanism at MGH and work with the FDA to get this IND approval. We've submitted our protocol to the scientific review committee, and we're at the stage now where we're working on revisions, and we hope to submit it to the IRB probably in about a month, and so then we would anticipate being able to open our clinical trial beginning of January 2015. So that's sort of our timeline going forward. So what are our goals of this project? This is really a pilot study, so we're really trying to generate some preliminary data. Nobody's really used this PET tracer before in brain tumors, and so we're trying to figure out if there's a signal there. Is this going to work in people who have brain tumors? So on one level, it's a high-risk, high-rewards project, because if it works, this would be a great solution for what is really a very clinically relevant question. Even if it's not as successful as we would hope, because we always have to prepare for that also, going through this process and really putting our team in place really helps us to optimize our technique. Meaning, even if it's not this PBR PET tracer, there are other PET tracers out there that are also equally interesting, different other novel imaging techniques. But having this pipeline, this team in place who have now gone through this process once, it'll make it much easier for us to do the next project down the road. Once we have our preliminary data, we can leverage this data and go apply to the NIH. the NIH always likes to see some preliminary data of success before they're going to fund you for a larger scale clinical trial. We also will, once we have this preliminary data in hand, we'll be able to go talk to pharmaceutical companies that are developing immune modulating therapies. Pharmaceutical companies love to have these imaging surrogate markers of response. Is their drug working or is their drug not working? And having an imaging tool that can give you that answer very quickly is a very useful tool for them. So we can go approach pharmaceutical companies and say, hey, look at this great test that we have. And then this also has implications beyond just primary brain tumors. It also has implications in the setting of metastatic brain cancer. So these are the tumors that start someplace else in the body and spread to the brain. So how can you help us further accomplish this project? So I think there are probably three main ways that an individual person can help us do this kind of research. So certainly one of the first one is getting to know your academic institution. So getting to know us at MGH and learning, there's so much fascinating research that we're doing. And so learning about the research that clinicians and scientists are doing at MGH is a great entry point. We love having people come visit us in our labs and showing off our cool tools and MRI scanners and different techniques, our cyclotron where we make the PET tracers, all these fun things. And so start with the doctors that you know. There's always the development office. They're always happy to help people as well and sort of match people up with what might be a particular interest of yours. But come talk to me, come talk to anybody, and we'd love to get people involved. The other option is the Cancer Center has patient advocacy groups, and so these groups work in the Cancer Center to develop different programs for patients and different ways of getting involved directly with the Cancer Center itself. And then the second possibility is to network with some of these regional or national patient advocacy groups. So for example, there's the National Brain Tumor Society. They're actually based in Waltham. And they work a lot to raise awareness about brain cancer. They work some with the FDA to encourage the FDA to approve drugs faster, work with the NIH to try to encourage funding of the NIH. But it's sort of a broader, maybe less intimate role than working with your local institution. And then finally, probably the biggest thing is really NIH funding. And so lobbying your congressman or congresswoman to support NIH funding is critical. The NIH budget has gone down significantly in the last several years, obviously with the recession. And so there's been a remarkable decrease in NIH funding over time, and it's really been flat in the last couple of years. And so applying for funding to get these clinical trials up and running is very difficult. So you need to write your congressman and say, fund the NIH, get more money for clinical research. And so I think these are some of the three key points that an individual person can do to help us. And really, ultimately our goal with all of this research is just keeping the patients at the center, because those are the ones who are really suffering through a very challenging, incurable type of cancer. And so what I try to do is I wear a hat of a clinician and a researcher, and so there's this nice circle that tries to support the patients. And so there's a lot of discussion between the patients and me and the researchers, and then our clinical trials team that really supports and runs these clinical trials to really advance the care of patients with brain tumors. But really our ultimate goal is to help individual patients and really optimize care for patients. And so this is my team. So I work a lot at the Martinez Center for Biomedical Imaging, which is a joint venture between MGH and MIT. And so this is some of the people in my lab. And so having these projects that these people get excited about is very important. So this guy has now gone on to graduate school at Harvard, And so he's going to be the next generation who is going to go on and develop the new imaging techniques that will help cancer patients. And so getting him involved early in some of these projects is, I think, a very important step to inspire the next generation to carry the torch forward. And this is just a list of the names of the people that I work with. We have a large clinical team. We actually work with some people in Norway, and then the Martino Center for Biomedical Imaging, where we're really sort of working to develop these novel imaging tools to better understand how drugs work, better optimize combination therapy, and really better understand tumor biology so that we can move the field forward. Thank you. Hopefully I didn't talk too fast. And I'm happy to take some questions.
I'd like to just inquire if you have an opinion about what had happened. One of them was a young man about 35 years of age who had, to the best of my knowledge, an inoperable brain tumor. They couldn't operate. The family pursued all kinds of alternative solutions to it. And within a year and a half, he steadily went downhill and finally died with 2 young boys in his family. The second one was a 20-year-old niece of mine who was diagnosed with a cancerous tumor. And from the time we heard about her having been diagnosed as a result of very severe and unusual headaches, we were quickly informed that she was going to be operated on within 3 days. And she was operated on within about 5 days. And she survived with apparently no difficulty. And I'm just curious about those outcomes as to why they were so extreme. We lost the young man, the young girl survived.
So I said generically brain cancer, but there are over 100 different types of primary brain tumor. So the prognosis and the outcome really depends on what the underlying cancer type is. So there are ones that can be surgically cured, and they tend to be in younger people, but the majority of them are not surgically curable, and they can be very aggressive and go on and end somebody's life. But it really depends on what the type of tumor it was that each individual patient had. The other challenge is the location in the brain. You know, location. Real estate is very key in the brain as well. And so if a tumor is not in an area that the patient can be operated on and have it taken out, then that also can suggest a worse outcome as well. So it really is that location and then underlying type of tumor that predicts how the tumors are going to behave.
Doctor, my question is specific to your clinical trial. Since you told us that with this specific type of cancer, there are relatively few patients, I think you said 10,000 in the country, what number of patients do you anticipate you would have participate in your clinical trial? And then, assuming that it's a relatively small number, I wondered, would you consider opening that to metastatic cancer patients? For example, you just talked a little bit about cancers that occur in the brainstem for which there are no other treatments available.
So the nice thing about being at MGH is we're a referral center for brain cancer, and so we see a lot of patients. Unfortunately, we see a lot of patients, but it also enables us to do this kind of research. So I think we were anticipating, if I remember correctly, about 30 patients in our clinical trial. So it'll be enough for us to see if there's really a signal there. And then when we go on and to do a larger clinical trial, we will open it up to enrollment at other sites. We did actually have an arm for metastatic melanoma in our trial, because melanoma patients are treated with a lot of immunotherapy, and it works fairly well in the brain, but not as well as melanoma in the body. and unfortunately, it boils down a little bit to a cost issue and how much MRI scans and PET scans are about $4,500 a scan. And so we're somewhat limited in how much we can do. We have a small cohort of metastatic melanoma patients in there, but it is definitely something that we would like to expand and do more of in the future.
We all want desperately to find answers to the questions you're searching for, but to what extent does the process really help? To what extent do you know what Anderson's doing? To what extent do you know what Mount Sinai is doing or any other hospital? And how do you coordinate this? To what extent do you share information to try to find an answer to these questions?
So there are different mechanisms to share information. So there is actually the Adult Brain Tumor Consortium, which is an NIH-funded consortium of different academic centers. And there are several clinical trials that are run through that. And so there's actually a lot of interaction in our community. We're also a smaller community, the neuro-oncology community. But even in other types of cancers, there are these different national consortiums where multiple trials are being run. And so there is some collaboration across sites. So we're a member, Sloan Kettering's a member, UCLA, UCSF, all these other ones are members. And there are 2 or 3 that run brain tumor clinical trials. And so in that sense, we do crosstalk and do collaborative work in that way. The NIH is pushing a lot more they will fund collaborative trials more than they will fund single institution trials. The challenge is you have to start somewhere. I can't go convince the Adult Brain Tumor Consortium to do this type of trial because they don't even know if it's going to work. So to try to get the 10 member sites of the Adult Brain Tumor Consortium to open this trial is going to be difficult because it's expensive. They have to be able to make the PET tracer. There are just some hurdles that they're not going to embark on unless there's some preliminary data to say that it's worthwhile to do. So all of these pilot studies are going to be done in single institutions. And then once you have some data, you can take it to these larger consortiums.
There's been a new conversation put on the front of People magazine, and everybody's talking about the 29-year-old who died 2 days ago. Are you familiar with that case?
You know, the frustrating thing about that is, she did have a glioblastoma. It sounded like it was a grade 4 tumor. The challenge is young people tend to do better than old people because they're older people, because they tend to be more healthy and stronger. And really our goal is not to make the treatment worse than the disease itself. And so it's frustrating as a clinician to see somebody make that decision to forego treatment when, everything that I strive for is to make this treatment preserve quality of life and make sure that it's not just quantity over quality. And so, but that fine line is very different for every individual person. And so for whatever reason, she just decided that it wasn't right for her. But we really don't want their treatment worse than the disease itself. I'm a firm believer that the treatment can extend quality of life and not just quantity of life. So it's hard to know in her individual case why she made that decision.
I'm interested in knowing a little more about the background of your choice in using imaging to try to analyze the effectiveness of the treatment. Are you indirectly saying that there are no chemical markers that have been found or no other viable means of understanding where you are in the treatment, that imaging is basically all that anybody's been able to figure out to do at this point?
So imaging really is the mainstay of how we assess tumor response. Really in most cancers, sometimes you can go back in and take another sample of the tumor out and look at it under the microscope and stain it to see how the treatment has been impacting the tumor itself. But in brain cancer, that's very difficult. It's hard to go back into the brain multiple times and just take a little sample out and see if your drug is working or not working and why it's working or not working. So in brain cancer, we're really limited to this non-invasive imaging tool to assess response. And imaging has really moved beyond just anatomical imaging. We get a lot of functional information as well. So we can look at blood flow, we can look at swelling, edema, scarring. It gives us a lot of information as to what is going on. We're doing a project where we're looking at circulating tumor cells, so seeing if you can draw a blood sample and measure tumor cells that are spilling into the blood. Again, that hasn't worked as ideally as we would have liked in brain cancer because the brain is the sanctuary site and it's protected by this blood-brain barrier. And so the tumor does not spill into the blood in the way it has in some other cancers like lung cancer or something like that. So in brain cancer, at this point, imaging has our best way of non-invasively and safely assessing response because you can do it multiple times without harming a patient. And so we'll find some other tools, but I think right now this is, sort of a good way of looking.
Okay. Thank you.